welcome our another eminent speaker, Dr. Pankaj K. Tyagi sir, who is currently working as a professor in Department of Biotechnology, NIET. Dr. Tyagi sir has done PhD specifically in Drosophila genetics from MD University Rohtak, and he has published more than 60 research articles in several reputed journals. He has also been the editor-in-chief of Biological Insight Journal and also member of editorial board of several reputed journals in the field of nanobiotechnology, Drosophila biology, zoology, botany, and toxicology. His research area includes nanomaterial synthesis from biological sources and their toxicity in Drosophila species. He has received numerous awards from such as University Research Fellowship, recommended UGC Research Award, and including Fellow Linnean Society, London Prestigious Award. In the field of nanobiotechnology and Drosophila biology, he has also obtained several research grants from various organizations, including Department of Biotechnology, Department of Science and Technology, UPCST, AKTU CRIP, and LSFI. He has also obtained grants for several workshops, conferences, and short-term training programs from various funding agencies. He has always been a great source of inspiration for all of us, specifically in the field of research. Hence, I would like to request our Professor Pankaj Tyagi sir to kindly enlighten us this event with your insightful lecture. Please, sir.
Thank you, Pratibha. You may introduce in a very nice way. Thank you, Pratibha, again.
Thank you, sir.
Okay. Okay. Dr. Arvind, can you confirm the PPT is visible?
Yes, sir. It is visible. अगर आप चाहें तो इसको स्क्रीन शेयर मोड उस पर रख दीजिए. स्लाइड शो
Yeah. Slideshow mode. Yeah, it is on slideshow mode now. Okay, now PPT is on slideshow mode. Can you confirm?
No, it is still in the same mode. Okay.
No. Okay. So good afternoon, all participants. And assuming that you have the little knowledge of nanobiotechnology. Okay, sorry for interrupt. Now, Pratibha, I am audible.
Yes, sir, you are audible.
Okay. So, this is the title of today's lecture: Nanobiotechnology and their application in precision agriculture. Okay, so assuming that some of the students have little knowledge of nanobiotechnology, I have seen the specialization of participant and the different backgrounds are the participant. So the basics of nanobiotechnology first discussed, and after that the advanced level, and then the applications of nanobiotechnology in precision agriculture. Okay, so this is the outline of my lecture, the introduction of the nanobiotechnology. This is the basic history and origin, fundamental concepts, how many approaches of nanobiotechnology, and that is the science behind that nanotechnology with nanomaterial dimensions. Why small is good? Surface area to volume ratio. This is the science for that behind the nanomaterials. Property of nanomaterials, different types of nanomaterials, and in the last, the applications of nanomaterials in precision agriculture. The first question, what is the nano? Nano is the subject, nano is the science, nano is the technology, and what is the nano? So I think this is a very misunderstanding of the students and some teachers also. Nano is a subject. So nano is a science, nano is a technology, and that particular technology in which we manipulated any matter on an atomic level, molecular level, or supermolecular scales level. So if we have any precision types of machines, such as an atomic force microscope or scanning probe microscopic, in which with the help of that particular machine, we change the atomic structure of any matter, that the technology is called nanotechnology. Okay, and this particular technology is not a new technology. It is very ancient, very old technology. And the first country which is accept that particular technology in the next US. US established a particular forum, a particular R&D department. The name of this department is National Nanotechnology Initiative. This is the US government R&D initiative, and that particular initiative was started at that time. The Bill Clinton is the president of the US in 2000. Okay, and now that particular department is the merger of the 30 departments and some independent agencies and some commissions work together and increase or infrastructure R&D divisions of the US country. Okay. According to the NNI, the definition of the nanotechnology as the manipulation of matter at least one dimension. One dimension means dimensions are three types: one dimension, two dimension, and three dimensions. So, nanomaterials fall under one dimension also, two dimension, and three dimensions. But the scale size of that particular atoms is 1 to 100 nanometers. So, what is the difference of one dimension, two dimension, and three dimensions? This will be discussed in the next PPT. Okay, so here are some old examples in which the thousands year ago Chinese used gold nanoparticles in the form of inorganic dye to color the ceramic porcelains, and some Romans and Greek used the nanoparticle to dye their hair. And what are the nanomaterials works when the dye of the hair? In hair is proteins present in the hair. They will be colored with the help of that particular dye. And when the lights come to hairs, they may reflect the different colors. So Roman and Greek persons use that particular dye to color the hair. And some Egypt, Egyptians were using nano lead to prepare makeup of the eye color. Okay, and these particular applications may be elaborated by 1857 Faraday when they work on the light versus color, and they may be prepared the gold colloid. If you see here, the same colors, same suspension of the gold, but when the light is transmitted, the color is different, and when the light is reflected through that particular tubes, the color is different. And the common examples in the Liker gas cup. Liker gas cup is a very ancient cup which is available in the British Museum in now, and very frequently used in the 4th century in the Rome. And what is the property of that particular glass? That particular glass is made up with the dispersed gold and silver colloidal particle on that particular glass. But what is the beauty of that particular glass? The beauty is when the light is passed behind that particular glass, they may be appearing the red in color, and when the light in front of that particular glass, the glass may be in green color. So the red is the transmitted light and green is the scattered light. And this will happen due to the property of the nanomaterials. And some ancient examples of the basmaj, and now they nowadays they frequently use of the basmaj to ayurveda, and some important basmaj is I think the gold as silver basm, hirat ratan jayanti basm, or such a types of bhasm. That particular bhasm may be prepared particular materials. For example, the gold material may be fabricated, micro-fabricated and then nano-fabricated at the level of nano. Nano means 1 billion part of a meter. Now comes to the history and origins. The early history and usage of the nanomaterials. If you see that particular beautiful picture here, this is the stained glass window, and that particular stained glass window frequently used in the 6th to 15th century in the Europe. And what is the beauty of that particular glass? The beauty is provided the photocatalytic air purifier and the use of the titanium nanomaterials at that times in 19th to 17th century, the glittering effects of the luster is used in the Islamic world or Islamic countries and later than the Europe. And what is property? They may be used the silver and copper or other metallic nanoparticles on that surface of this ball. And here is a very, very important examples, the 13th to 18th century, the Damascus steel. Damascus steel is a very, very important steel and the main origin of that steel in the southern part of the India. And the manufacturing the that particular materials to the Sri Lanka. So the sword of Tipu Sultan is made of that particular steel, Damascus steel. What is the beauty of this steel? The nanotubes and cementine nanowires present of that particular steel, and ultra high carbon steel formation that give them strength, resiliency, and ability to hold keen edge. If you see this picture, this is the tan image of that particular steel. So uh, the very, very keen, very, very re-silencing and strengthful weapons prepared through that particular steel. What is the conceptual origin? Uh, can any evidence is present in the science? What is the era and which particular year in which the conceptual origin is start? Yes, Richard Feynman. Richard Feynman is the example of that particular questions. The conceptual origin may be start in the December 1959. And if American Physical Society meetings, they may uh, deliver a lecture. That is the novel lectures. And the title of that particular lecture, here's plenty of room at the bottom. What is the meaning? What is the conclusion of this lecture? In this lecture, Richard Feynman emphasizes that if we prepare any precising machine, with the help of that machine, we may write the 24 volumes of Encyclopedia Britannica on the head of a pin, or an encyclopedia may be written in the grain of rice. At that time, the pupils not believe in what says the Richard Feynman. But when the AFM and STM is invented, this is possible. Norio Taniguchi, another milestone in the field of nanotechnology in 1974. In a conference, the Japanese professor of Tokyo University, Norio Taniguchi, first time introduced the term of nanotechnology. So he is the first scientist to use the term of nanotechnology in his paper. This is the A. Eric Drexler. A. Eric Drexler the popularized the nanobiotechnology. The whole credit goes to the nanotechnology for popularization goes to Dr. K. Eric Drexler. Dr. K. Eric Drexler promoted the technological significance of the nanoscale phenomena through species and writing some books. The one book is very popular, Unbounding the Future. And the second book is the engines of creation, the coming era of nanotechnology. And the era of that particular A. Eric Drexler is 1980 to 1986. He is also start a private institute. The name of that institute is the Foresight Institute, but he is no run is no more because that particular institute not getting any funding. But they may be popularized nanotechnology. Without any experimental research, without any experimental research and advancements, no technology may be popularized. So what is the need? Need means how to popularize as a practical basis that particular subject. So if you are aware, please wait 1 minute. Okay. If you see here that in 1960s, the transistors may be used. Transistors may be used. Mostly transistors may be used. And the transistors is made up of the gate oxide and thin films. But when the nanotechnology and nanoscience got boost in early 1980s with the two major developments. First major development on that particular is the cluster science, and second is the inventing of scanning tunneling microscopes. So when the comes in the market cluster science and scanning tunneling microscope, the MOSFET may be the used in the form of gate oxide and thin film. So MOSFET is a small chip based transistors. Okay, after that, the two inventions, the scan probing microscopy, STM developed in 1981 and AFM is developed in 1986. So I think some of participants may be aware that the 1981, the STM invented by the Gerd Binning, Henry Schroer, who is the employee of IBM Zurich Laboratory and awarded the Nobel Prize in the year of 1986 in the field of physics, and in which year they may be awarded the Nobel Prize in STM development, also may be invented EFM, and the one scientist is the common Binning. The other scientist is the Quat and Gerber, who is the invented EFM, and they are also commercialized the AFM in the market in 1989. And here is another very important thing that if you see the IBM logo developed by the Don Igler, Don Igler. This is the IBM researcher, है ना? And Don Igler maybe used the 35 genome atoms to manipulate it. After manipulated and spell out the IBM logo. Okay, the another advancement of the interface and colloidal science to discover the Fuller family. Fuller family is related to the carbon, C60 family. And C60 family may be discovered by the Harry Porota, Richard Smalley, and they may be get the Nobel Prize of the Chemistry in 1996. So after invention of the AFM, STM, scan probing microscope, colloidal science, and Fuller family, nanobiotechnology is very easy and popularized for the practical aspect. But without controversy, no may be the popular. Without controversy, the popularity is not getting. So some scientists and some writers maybe written the articles. The one most important articles is the written by the Bill Joy. He is the chief scientist of Sun Microsystem, and the magazine in which the written that particular article is Wired magazine. And in which particular article they may be this technology is not useful to humans. This technology may be create a dominance on the machines. And after that, the Michael Crichton write a novel Prey. And if you know, the some movies also uh, uh, developed in the that particular theme. For example, the Terminator. Judgment Day, Terminator 3, Rise of the Machines, based on that particular novel. And after that, the Royal Society of, please wait 1 minute.
हाँ जी।
Okay. Uh, Royal Society uh, released a report in which the toxicological aspect of that particular science may be released. That what are the toxicological implications? Maybe this technology. Uh, and this is the some examples, some initial commercialization applications of this technology. Without commercialization of any technology, not a popular for the pupils or the country. So here, if you see the consumer applications, the shirt, some beauty products, and if you see in the TV, I think the daily 3 and 5 advertise maybe the belongs to that particular technology. If you see the Samsung machines, the dryer and the washing chambers is coated with the silver nanoparticles. And that particular silver nanoparticles, the ability not contaminated the cloth with the bacteria. Well, and here some locks, locks are also coated with the gold nanoparticles. Some medical products, beauty products, sporting products, and uh, daily usage products use nanotechnology, especially the zinc widely used in the paint. Zinc used in the paint. After that, what is the fundamental concept behind this technology? So the fundamental concept is the 1 nanometer is 1 billionth part of a meter, or 10 to the power minus 9 of a meter. By comparison, the carbon-carbon bond means the carbon-carbon is a covalent bond. So the covalent bond are the range of 0.12 to 0.15 nanometer. If you see the DNA double helix structure, so DNA double helix structure at a diameter around 20 angstrom. But if that particular diameters comparison into the nanoscales, that may be covered 2 nanometers. And on the other hand, the smallest cellular life form of the bacteria, genus Mycoplasma, exists in the earth is 20 nanometers in the length. And by conversion or conventional nanotechnology, the real definitions which is provided by the National Nanotechnology Initiative by US Department, the any material having the range 1 to 100 nanometer is called nano. Okay, so how many approaches of the nanotechnology? Basically, four type of approaches exist to study the nanotechnology. The first is the bottom-up approach, second is top-down approach, third is biomimic approach, and fourth is functional approach. But the two approaches most frequently used as a nanotechnologist. Okay, the first is bottom-up, second is top-down. What is the difference in both approaches? Difference is that if you prepare the nanoparticles or precising atoms to use the bulk materials. So larger ones may be fabricated into smaller ones. That is the top down. And that reverse, if the smaller ones may be created any device which is the larger, that is the bottom up. So if you see in the examples of nanoparticle synthesis, here a bulk material with the top-down approaches, they may be fabricated, nano-fabricated, maybe fabrication process may be the physical, fabrication process may be the chemical, and fabrication process may be the biologicals. They may be fabricated into the small powders, and that particular small powders may be created nano structures, monolayers, and that nano structure may be converted into nanoparticles. That particular nanoparticles may be organic nanoparticles in the form of carbon 360 family members. They may be the fuller, they may be the diamond, graphene, nanotubes, buckyballs, or they may be the metallic nanoparticles, the silver, gold, cobalt, nickels, and the second way, the bottom-up approach. Bottom-up approach maybe start the precursor. For example, if you prepare the silver nanoparticle, here the precursor is the silver nitrate. So silver nitrate is work as a precursor, and they may be ionizations by the energy and formation of ions by the chemical methods or biological methods as a free radicals and make a clusters. These clusters may be converted in the nanoparticles. So these two approaches are frequently used in the field of nanotechnology, bottom up and top down approach. Now, comparison of macro, micro and nano. So here is a bit clear. Macro means the phenotypically, if you see phenotypically any persons as a human beings, the height is 5 feet and 6 feet. It is a 2 million nanometers. If the convert into the nanoscales, the size of the apple is the 8 centimeter, but when the apple size is convert into the nano, that is the 80 billions. And the same having in that particular ant the 5 million nanometers. Micro, which is not visible naked eye, that particular material visible under the microscope, because the human eye cannot see 10,000 nanometer less. 10,000 nanometer less may be visible to see the microscopes. So if you see here the diameters of the human hair, which is the 75,000 nanometers, and the bacteria, some viruses, and other structures maybe through microscopes. That is the micro. And nano. Nano never seen with the simple light microscope and not a naked eye. Nano visible through the scanning probe microscopy. Here, if you see that buckyballs, if you see the DNA, which is the 2 nanometer in size, and the carbon nanotubes having with the size of 1.3 nanometers. And the lowest nanomaterial size is the hydrogen atom, having 0.1 nanometer in the size, hydrogen atom, the lowest. Simply, as a layman, ask the question how to define the nano. So you may be simple. Suppose this is the map of the India, and the population of the India is 1 crore. I just suppose, not a 1 crore. I think the population of the 1.35. But if the population is 100 crores, so one pupil, you and me, as in nano, with comparison the total population of the India. Or another examples related to the money. If you have the 100 crores rupees, the 1 rupee is the nano as compared that particular 100 rupees. Now, I think when the start my presentations, I discuss the three terms: one dimension, two dimension, and three dimensions. So here, the difference is what is the one dimension, two dimensions, and three dimensions. One dimensions, any materials which has only one parameter, either length, either width, or height. That is the one dimension. But the material under falls in the nanoscale. And what is the nanoscale? Nanoscale is the 1 to 100 nanometer. So if any materials having the scale of nanometers means less than 100 nanomaterial and cover only one parameter, that parameter may be the length, width or height, it may be the one dimension. And one dimensions is the thin films, layer and coatings. These three may be used the fabrication of most fixed chips or chip dimensions. Two dimensions may be cover the two length or breadth. But again, The materials which has the two dimensions cover the nanoscale, less than 100 nanometers. So this may be the examples of nanotubes, nanofibers, and nanowires. Three dimension. Three dimension, it has all parameters. All parameter means length, width, and height. The three things is present any materials, but under fall the nanoscales 1 to 100 nanometers come to the three-dimensional nanometers, and that is the example is the nanoparticles, nanocell, nanorings. Why small is better? This is the very important questions. Why small is better? Small is better due to the surface area. When the surface area increases, the size is decreases. If you see here, uh, one minute please. If you see here, this is a structure, one cell structure. In this one cell structure, the total number of atoms is 13. When the structure is increases, the second structure is the two cell structure. It means the number of atoms is increased. If we increase cell by cell this structure, and here the structure is the 7 cells, means the atoms is higher, but the surface size is the lower. So when the surface size is higher, here is the surface size of higher, means the number of atoms is very low and the cell is singles. So small things, larger surface size may be the faster, they may be the lighter, may be the cheaper, more efficient, and work properly. But what is the behind that particular smaller size? This is the surface area. So here, if you see surface area, and it's no blue. Okay, the surface area. Surface area is equal to height into width and number of the sides and number of cubes. So here, this is the cube. That particular cube into the height 2 millimeters and the width is 2 millimeter. And number of the cube is 1. And the how many sides of this cube? The 6 side of this cube. So come to this point. The surface area height 2 into width 2, number of sides 6, and number of cube is 1. So that is the 24 millimeters. But if we fabricate that particular cube into eight eight types. So here the cube, now the cube is the 1 millimeter size. Then the 1 millimeter height of the cube, 1 millimeter width, and number of side for each cube is the 6, and number of cubes 8. This is the 48. Can, when the surface area is changed, can the volume is changed? Come to this point. What is the volume? Volume height into width into length and number of cube. So we see here the width of the cube is the 2 mm, height is 2 mm, length is 2 mm, and the number of cube 1. So volume is 8. And here the width, height, and length is a 1 and number of cube is the 8. Volume is 8. So no change of the volume. But if we miniature the atoms or miniature the things into the smaller form, they may be increase the surface area. So if we see here, the surface area oblique volume ratio is the 3 and just double the surface area oblique volume ratio. Uh, what is the science behind that particular nanofabrication? Nanofabrication, if any cubes is the fabricated into the small pieces, means the hidden surface area exposed to the materials. Okay, if your materials may be the larger size, so some sides of that particular material may be hidden. But when the fabricated into small pieces, so each and every side may be exposed to the material. Now comes to the property of the nanomaterial. The property of materials can be different at nanoscale into two main regions. The first region, nanomaterials have relatively larger surface area, okay, as compared to the mass materials or the bulk materials. And second, the chemical, the small things or nanoparticles chemically more reactive as compared to the bulk materials. Second, the quantum effects can begin to dominate the behavior of matter at the nanoscale level. Okay, origin of the property. If you see the bulk material here, the two types of the states, one in unoccupied state, second in occupied state, here the conduction band and valence band. No gap between these two bands when the material in the bulk. But when the decreasing the size and comes to the nano, a, a gap is appearing between the conduction band and valence band. And that particular gap, electrons maybe motions become confined and quantizing sides. And that separation between the valence and conduction bands make this material more reactive. Here, this is a very beautiful examples of the gold materials. You see that the bulk gold in the yellow color, and bulk gold is the inert material, shiny yellow color. But when you fabricated the gold into gold nanoparticles, it may be the different colors. If you nano fabricated into 500 nanometers, this property is metallic, turbid, and blue in color. If you precising or manipulating any precise, means the color may be violet. And if you fabricated the 100 nanoscales, means nanoscale 1 to 100, that gold nanoparticle in metallic and red in color. Okay, but if you fabricated on 1 nanometer scale, that the property change of the gold metallic to non-metallic and orange in color. And if you help of the some precising machines may be fabricated as a atomic levels, that may be the colorless. So this is the beauty of gold nanoparticles. Gold nanoparticle may be inert when in the form of bulk, they may be metallic when in the form of nano, and they may be non-metallic when in the form of the quantum, and it is colorless or without any property when in form of the atom. Nanoscale size effects. Nanoscale size effects, they may be increasing physical property. They may increase the chemical property in the form of reactivity, physical property in the form of metallic point, electrical property in the form of conductivity, and mechanical property in the form of strength, and optical property, the light emissions. And if any synthesized nanomaterial having the metallic property, so they may be checked through that particular exercise. This is the metal and suspension of colloidal suspension of the nanomaterial. That nanomaterials may be comes to the magnet side. Different types of nanomaterials. Some nanomaterials natural occurring in the form of forest fire, in the form of sea spray, mineral composites, volcanoes, and viruses. But some nanoparticles man-made, and that particular man-made nanoparticles are two types. First is incidentally. For examples, the cooking smoke, diesel exhaust, welding fumes, industrial effluents, and instant blastings. So incidentally form the nano materials or nanoparticles. Second, then incidentally nanoparticles have more variable. It means they have the different shape. They may be the spherical shape, rectangular shape, rod shapes, triangulars, but having the size of nanoscale. And some nanoparticles are engineered nanoparticles. Engineered nanoparticles may be more well defined. For example, you may you may surface ligand based that particular nanomaterials this the cell and this the core. So you maybe change this core and cells and maybe functionalize to different antibodies, different groups, and maybe coated to organic coatings. And human origin engineered nanoparticles, maybe the metallic nanoparticles, maybe quantum dots, buckyballs, nanotubes, means carbons, nanoparticles, sunscreens, pigments, and nanocapsules. Now comes to the overview of nanoparticles. If you see the size of the nanomaterials, the size of the nanomaterials is the 1 nanometer to 100 nanometers. So all materials having with that particular scale 1 nanometer to 100 nanometers is called nanomaterials. Now come to the shape. Shape of the nanoparticles is spherical, cubic, rod, star, plate, and sphericals also. These are the shapes of the nanoparticles. The materials. Materials, nanoparticles may be the metal nanoparticles may be the gold, maybe silver, zinc, carbon, copper, sorry, and organic nanomaterials may be the related to carbon family, carbon nanotubes, dendrimers, protein drugs conjugates, liposomes, and polymer particles. These all things may use as a drug carriers. Surface is a very important. Nanoparticle surface may be surface functionalization through PEG, through surface functional group, through surface charges, and through target ligands. That particular target ligands will be antibody, peptidal, and aptamers. Now come to the applications of the nanotechnology in precision agriculture. First, what is the precision agriculture? Precision agriculture means the approach of any farm management or agriculture to use the information technology or computer science technology to ensure crop and soil both receive exactly what they need for optimum health and productivity. Means the goal of the precision agriculture to ensure profitability, sustainability, and protection of the environment. This is the precision agriculture. So if you see this figure mm, here, so first the precision agriculture or agriculture farm may be divided into three parts. First, the data collection part. Second, interpretation, and third is the application. So data collection part is very important. If you start the precision agriculture or preparation of the farm, first you maybe collect the pictures of any other farms, the map your farm soil, crop conditions map, soil conditions map, and yield map. So these are the other farms soil. This map is the other crop conditions map. This map is the soil condition map for the other farms, and this is the yielding map of the other farms. So first the collection of data. This may be same as when you start your thesis preparation of PhD, start the PhD. So you collect the research papers and on the different topics and matching that particular research paper according to your need. When the all collected data related to the crop and soil may be interpreting or putting in the PC, which is the part of the IT or computer science department. And the better results may be the output of that particular field. And this is the treatment map. So you need that particular map. If you have this type of soil property, if you have this type of crop conditions, if you have that particular conditions of the soil, then definitely you may get the yield, better yield. So after interpreting that particular map, you may apply in which field when you start your farm. Means the cultivated with the help of that particular automatic machines, the levels of the land is equal without driverless tractors, and after that the fertilizer. That particular fertilizer is not sprayed manually because the manual spray of fertilizer may be somewhere the higher amount of the fertilizer and somewhere is the less amount. So the particular map, you map the equal distribution of the fertilizer with the help of that particular automated machines. And after that, the seeding, proper seeding of that farm. This is the product. A future smart farm. If you see what is the future smart farm with having that precision agriculture, so you may see the cloud storage. Cloud storage is also help you to matching the data. So any times you match your data when you need. You may require a drone. Drone have a fly over the fields locating weeds where the weeds conditions is more, pathogens conditions monitoring, and the animals, which animal is a sick animals, you may be out from the healthy animals in the group, and collect data about the development of the crop and their needs. So the needs of drone is very, very important in the smart farm. And smart collars. Smart collars is the important things. The cattle and buffaloes present in the smart farm have uh, that smart collars on cattle send information about their health and any other data that may be used in tests such as for interest when when an animal is ready to give birth, or if any animals go so far away from the farmhouse, so you may be detect through that particular smart collars. And if any one persons uh, maybe harm your animals, the smart collar you intimate firstly. Third is the smart crop sensor. Smart crop sensor collect data about the moisture, about lack of nutrients, of the presence of pests to determine the need of irrigations, fertilizer, pesticides, and the right time of the water irrigations. The automatic system, irrigate system may be automatic. Okay, the fertilizer and fumigates each plots depending on the individual needs. And self-driving vehicles so with precisions and plant seeds at exactly the right depth and separation of the type of crop, maximizing the output of the land. The benefit of the smart farming. What are the benefit of the smart farming? The smart farming may be beneficial for increased production, water saving, better quality, reduce cost, pest detection and animal health, and better sustainability. The increased production, the optimization of all the process related to agriculture and livestock rearing increased production rate. Okay, water saving, weather forecast and sensor that measure soil moisture mean watering only when necessary and for the right length of time. the better quality. Analysis the quality of the produce obtained in relation to the strategy applied makes adjustment possible to increase subsequently production quality. Pest detection. Pest detection means early detection of the infections, maybe detection of infections in crops and sickness in animals means that their impact on productions can be minimized and animal welfare improved. Better sustainability, saving resources like irrigations, water. Here, the how to nanomaterials versus precision agriculture can through this particular technology, nanobiotechnology, can we put some uh, into the precision agriculture? Yes. So this is the, I think all of you are aware what are the nano materials, nanoparticles having the size of 1 nanometer to 100 nanometers. These are the shapes and the nanoparticles, material in the plant science, some carbon-based nanoparticles, some metallic-based nanoparticles, some metal oxides, quantum dots, silicon-based and polymer. These are the nanomaterials. So how to protect their agriculture with the help of that nanomaterial, because the nanomaterials having the good property of to killing the bacteria, that particular bacteria which is the pathogenic in nature, that nanomaterial have the ability to kill the pathogenic fungus also. So fungus also. But do you know the what are the proper way to penetrate that particular nanomaterials or to spray the particular nanomaterials in which way? So these are the three ways for penetrations the nanomaterials into the plant body. The first is the foliar entry. Foliar entry means you may spray that particular nanomaterials on the plant leaves and stem. Second entry is the root entry. Root entry through irrigations. Uh, when you may spray that particular nanomaterials into the plant leaves, they may be entering into two ways. First is the cuticle, second is the stoma, and root entry through the xylem, and third is the plant tissue culture. So nanomaterial in medium, nanomaterial mixed into the medium, that particular medium which is provided the tissue culture materials. Here, this is the very fantastic examples of the copper nanoparticles, and that experiment is also done in our laboratory. Also, we prepare the copper nanoparticle and spray the plant, and that particular plant infected with the bacterial condition, they may be to improve these conditions after spray. And this is the fungal infection. When the copper nanoparticle is spray on the leaf, they may be improve the fungal infections. And also, if you add some hormones in the copper nanoparticle spray, the plant growth promotions and plant may be the healthy. But the entry of copper nanoparticles into foliar spray is very high, and through xylem is very low due to the size. It is a size related. Uh, Here again, the foliar entry through the phloem tissue and root entry through the xylem tissue. This is the very, very interesting. Uh, if Arvind sir give me permission, can we take more time?
Yes, sir. Yes, sir. You can continue.
This is a very, very important slide. Uh, Here, this particular picture divided into four types. First is the A nanoparticle trait. Nanoparticle trait means the nanoparticle characters. Second is the plant uptake. Third is the movement through the plant, and fourth is the penetration into the plant cell. So here, the nanoparticle trait. What is the nanoparticle traits? Nanoparticle traits in the characteristics of the nano. That particular characteristics is very, very important. Very, very important if we use into the plant size. First, the size of the nanoparticle. The first characteristic is size of the nanoparticles. The size of the nanoparticles is the mostly reports is their size is the 40 to 50 nanometer size nanoparticles easily uptake and move to the inside of the plant. The second is the types of nanoparticle synthesis. Nanoparticle synthesis. Nanoparticle may be synthesized through chemical, nanoparticle may be synthesized through biological, and nanoparticle may be synthesized through physical methods. So here, the best nanoparticles which is synthesized through biological process means the carbon source. They may be the easily penetrate to the plant system and easily move inside the plant. Third, the upper coating. Upper coating means the functionalization. Functionalization. How to functionalize the nanoparticles? To greatly change of the property. Functionalization greatly change the property of the nanomaterials. Okay, and again, this is the first. The second factor is the plant physiology. Plant physiology again the very, very important because the different species of plant may be showing the different physiology. Some plants are dicotyledons, some plants are monocotyledons, means the different types of the physiology. Third is the interaction environment. What are the environment in which you interact the nanoparticles into the plant media? It is very important. For example, uh, if if you are mix the nanomaterials into the water irrigations, so humic acids and organic matter is in the soil can lead to an improved stability and hence better availability of the nanomaterials. Whereas salt ion, salt ion might induce precipitations and trigger a contradictory effect. Even more the presence of other organisms. For example, the bacteria, fungi influences the plant uptakes. Okay, so this is the nanoparticle trait. Now comes to the plant uptake. Plant uptake, the foliar uptake. Plant uptake means the foliar uptake. Foliar uptake or plant uptake into two ways. The first is the lipophilic and second is the hydrophilic. So two type of uptake of the nanomaterials into the plant, lipophilic and hydrophilic. Lipophilic through diffusion, nano, nanoparticles may be diffused into cuticle with the help of the cuticle wax. And in the lipophilic, hydrophilic matter, the nanomaterials may be penetrate into the plant through stoma, yeah, stomata. What happens? The size. This is difference between the size. The cuticular size, cuticular pore size is the 2 nanometers. So if you manipulated nanomaterial or nanofabricated nanomaterial less than 5 nanometers, then the nanomaterials may be penetrate through cuticle. Otherwise, if the size is larger, so nanoparticle does not penetrate through cuticle, they may be penetrate through the stroma because the size of stroma is above than 10 nanometers. This is very, very important things to hear. The plant uptake procedure should be clear if you treat the plant. The second, if you see here the movement through the plant. So here two types of pathway, apoplastic pathway and symplastic pathway. Apoplastic pathway and symplastic pathway. में क्या difference? What is the difference between two pathway? The first pathway is apoplastic pathway. Transport take place outside the plasma membrane. This is the nanomaterials, and that particular nanomaterial transported through plasma membrane through extracellular spaces between them. extracellular, this is the extracellular space. Extracellular space between them, cell wall of adjacent and go to directly the xylem. When the nanoparticles reach it to the xylem, after reaching the xylem or cylindrical structure of the vascular bundles, through ascent of sap, the nanomaterials migrate through water into the whole plant. This is the apoplast pathway. Symplast pathway means the involvement of water and substance between the cytoplasm. Involvement of the water and substance between the cytoplasm of adjacent cell through specialized structure called plasmodesmata and sieve plates. Sieve plates are the part of the phloem tissue. And when the nanomaterial comes to here with the help of companion tubes and phloem, sieve tube, sieve plate, they may be distributed into the whole plant. I think this is the clear. Now comes to the third, penetrations of the plant cell. How could plant cell penetrations? If you see here, this is the penetrations into the plant cells. So here first is the endocytosis. Endocytosis. What is the endocytosis? This is the interaction of nanomaterials with plant cells. Endocytosis. The nanomaterials are incorporated incorporated into the cell by invasiations of the plasma membrane. Originate a vesicle that can travel to different compartment of the cell. Second, the pore formation. Pore formation. Here, the pore formation. Some nanomaterials can disturb the plasma membrane, including the formation of pore for crossing into the cell and reaching directly to cytosol without being encapsulated in any organelle. And third is the carrier protein. Nanoparticles can bind to surrounding proteins, including cell membrane, proteins that could act as carrier into internalization and uptake inside the cell. And third is the plasmodesmata. Plasmodesmata, another way of nanomaterial tube, entering the cell tube through plasmodesmata, specialized structure for transport between the cell. And now some scientists propose a different way, that is the ion channels. But that ion channels is the proposed way. No scientific study was done to yet. Here is a mechanism how to kill the nanomaterial to the bacteria or the fungal cell. So this is the hypothetic manner when the nanomaterials maybe first the nanomaterials line spin on the bacterial cell wall and uh, here after line spinning they may be create the electroporation. Due to the electroporations, the bacterial cell membrane may be disturbed and nanomaterial may be penetrate to the bacterial cell. After entering the bacterial cell wall, they may be induced ROS accumulations through ROS creating the cell death process may be stimulated, may be react with the DNA phosphate bonds, break the phosphate bonds, DNA damage, protein damage, also and ribosomes react with the ribosome and stop the protein formation, resulting the cell death. This is the bacterial cell wall, and the same happens with the fungal cell wall. In fungal cell wall, they may be react with the ribosomes and stop the protein formations. And in the some reports, the protein denatures also. And here react with the mitochondria with the cytochrome C and release the caspase again promoting the cell death and DNA fragmentation. So due to this phenomena, the nanomaterial is the best alternate to kill the bacteria and fungal pathogens. I think this is the link for today quiz, and this particular link may be sent to all participants. Dr. Arvind Kumar Arya. Thanks, sir.
Thank you, sir, for such a detailed presentation on nanotechnology and its application in precision agriculture. I think there is nothing more precise than nanoparticles or even the nanotechnology. So, at least for this session, we can ask participants to ask their queries. So, I am letting them in the meeting with their mics and videos so that they will be able to participate in the discussion. So here I request participants to raise their queries or any discussion they want to have here. Are we having any questions from the participants? Okay, I think the last instruction from Dr. Okay, Ajit sir, he wants to say something.
Hello, sir.
Yes, sir.
Ajit Kumar, I'm working nanotechnology. The nanoparticle which we use for land is affect people which we carry after the cultivation of land.
I think
Ajit sir, I think
uh,
Pankaj sir would have got your question, but your voice is little shaking.
Arvind sir, voice is not clear. It is better if participant write the question in chat box.
Hello, is it audible, my voice? Hello?
Yes,
Yes,
yes.
and now it is audible.
Okay, I'm asking the question which when we use nanoparticle like metal, like nanoparticle which we are using means that particle will affect the person which carry the food after cultivating.
No, 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 sir. This is the depend from the accumulation of the particle. If your particle is the non-toxic, if you prepare the nanomaterial with the biological pathway in which the minimize
Okay,
the
okay,
okay,
okay, sir. Thank you so much, sir.
welcome.
It was a valid question. I think many people are curious to know the toxicity related to a nanoparticle, and I would like to say this to all the participants that we have, like in the morning also, I said that we have state-of-art laboratory facilities here to test the toxicity of nanoparticles. And for this, we have Drosophila lab here, and we used to test the toxicity of these nanoparticles, especially first on Drosophila, and it actually mimics the system of human beings. So whatever toxicity they are causing to Drosophila, expected toxicity they would have caused to the human being also. So first it is tested on Drosophila flies, and then we can actually come to a conclusion regarding the toxicity related issues of these nanoparticles. Thank you, Ajay sir, for asking this question. So I'm still waiting for other participants if they have any query. Or otherwise, if you have any thing to discuss here.
Arvind sir, you also discussed the quiz.
Okay, there is a quiz at the end of the presentation. Dr. Pankaj Tyagi has shown the quiz link. So that link I will be sharing in the chat box, not in the chat box, but I think I prefer to preferably will be sending it through mail to all the participants. So you will be having a time till tomorrow before the session begins, like before the 10 p.m. You can attempt that quiz, and it will also give us your attendance in that session. Okay. So those who like all of you will be receiving the link. So kindly attempt it as soon as possible, and the answers will automatically be flashed after you finish the quiz. So it will going to be easier for you to attempt that quiz if you have gone through the lecture here. And one more thing we can do, we can also run the questions that you are finding in the quiz so that you will get familiar with the pattern the quiz is being asked. I think I can request this to Pankaj sir, at least play the quiz here so that the student can actually see the questions, and if they have any query then they can ask it to you so that they will get they get the clear concept regarding the nanotechnology and the thing that you have discussed in the lecture. So can we do this, sir? Can we play the quiz on the screen
Yes.
and they can see
No.
the questions so that
Yes, yes.
at least for the purpose of attempting the quiz they can raise the queries because
Yes.
otherwise nobody is interested in raising the queries.
Okay. Okay. Please wait.
Sir, Utkarsh Kaushik, guest, someone from the guest, he
Okay,
wants to ask a
yeah,
question.
yeah, I got him. Utkarsh sir, do you have any query here?
Yes, sir. Am I audible?
Yes, sir, you're audible.
Sir, my question is that sir has said about the nanotechnology and nanoparticle. So, is there any LD50 associated with the use of nanoparticle in the case of plants?
No.
Sorry, sir, I'm not able to get the answer.
Sorry, even I am not able to get the question. Okay, Pankaj sir might have got this. So Pankaj sir, will you please elaborate what the sir has asked and what means it?
Can, can you repeat the question,
Yes,
Mr.
sir. Yes,
Uttar?
sir. So, my question is that is there any LD50 for the use of
Okay,
nanoparticle in the case of
okay.
plant?
Okay, I got it. He's asking for the lethal dose like
Yeah.
LD50 we used to have in case of the drugs.
You may be calculated as LT50. Yes, you may be calculated.
Okay, I think
Yes, sir. I'm asking, is there any standard range is being mentioned for the different type of nanoparticles? Say, for example, if I'm
Sir,
using the silver, after this much dose, it will be going to be lethal for the plants.
you may be optimized in the laboratory after treatment the any
Okay.
in vivo study.
It's just like a hit and trial, just that we have to go for.
No, not a hit and trial. You may be optimize the
Then,
proper PPM, which PPM is not harm to any in vivo condition organism. For example, the rat, mouse, and Drosophila. So first, that particular
yes,
dose you may be provided to that particular animal, and after getting the
yes,
good
sir.
result, then you apply in the plant system.
Yes, sir. Actually, sir, I have done my nanotoxicology work on the male albino rats. At that
Yeah.
time, also we have studied the literature. So I thought ki in the case of plant, it is the same way or it is a different way.
No. First, you maybe optimize your drugs on that particular rat model, and after you calculate the toxicity, LC50 and
Yes.
LC100, then you may be applying the plant system.
Okay, sir. Because in the case of the animals, it is not been mentioned any LD50 in any of the standard literature.
You may be create, no problem. In the fly system is
Yes.
available.
Okay, sir.
In the fly system is
Okay.
available. Okay,
Okay. Thank you so much, sir. Thank you.
welcome. Arvind sir, the quiz is
yes,
visible.
yes, sir. The quiz is
Uh,
visible. I think all the participants can at least read these questions which you are going to attempt
okay,
in quiz. So, sir is only going to scroll down the quiz so that
okay,
everyone can see these question. And again, it is a request if anybody is having any query related to the question or the quiz, so they can come up with their queries.
okay. So if you see the first question, the size of the nanomaterial is between nanometer. Here the four questions, so you may pick the right. The diameter of the hydrogen atom. All question, all question in your presentation. Carbon atom make type or bond with other carbon atoms. So which type of bond? I think that particular bond name uh, discussed in your presentation also. Full range or buckyball is made up of carbon atoms. Carbon atom means it may be the C100, C75, C20 or C60 family members or others. Tears is plenty of room at the bottom. This statement was given by which scientist? Who coined the term? This is not a word. Word is also clear. Who coined the word nanotechnology? Name of the scientist. Egyptians were using to prepare makeup of pie. This is the nano aluminium, nano copper, nano steel, or nano lead. The soul of Tipu Sultan was made of, in which particular option may be correct? The width of the DNA molecules is how many nanometer? Greeks and Romans had used nanoparticles in the manufacture of. ये मैं बता, this will be discussed. This was discussed in presentation. Unbounding the Future book was written by which scientist? Fullerenes was discovered by name of the scientist. Who was the first scientist to describe the substance having nano dimensions process, although different and unique property? Okay, so this particular unique property is related to the gold nanoparticles and light experiment done into 1857. So you may be right, that particular scientist. In precision agriculture, the main role of which field? Agriculture, information technology, microbiology, or its involved arrangement of atoms? These are the two questions related to the surface and volume ratio. So you may be calculate. This is very easy questions. Both question is very easy. The full name of NNI. NNI a R&D department established by the US. Smallest cellular life form in bacteria and genus of microplasma are around nanometer in the length. STM was developed in which year? And this is the example in which we create a smaller device by using the larger one. This particular topic statements fall under the bottom-up, top-down, biomimic or functional approach. Okay.
Okay, thank you so much, sir, for showing us the quiz here. So we have in total 23 questions in this quiz, and all the participants are expected to answer these questions and the whole quiz before the 10 a.m. tomorrow. So I think that will be the fine time. Uh, Again, a humble request to all the participants, since many participants are joining right now in the meeting. So I think it will be better if they join the meeting on time, because our schedule is being shared already through mail to all these with all of you. So again, it is a humble request to kindly join on time, because otherwise you will miss lots of information that for which we are making so much effort from our side. Okay. Again, it is my duty to ask you here if anyone is having any query.
Utkarsh Kaushik.
No, he has already asked. I think he has.
Yeah.
Sir, I have a question.
Okay, Utkarsh. Yes.
Sir, is these questions are going to be shared or through mail?
Yes. Arvind sir, please reply. You are muted. Arvind sir, muted. Unmute yourself.
Okay. Yes, Utkarsh sir. I am going to send you the link of this quiz on mail. Okay. So soon you will be finding this mail. Okay. Any
sir.
other query you have? Like you want some clarification, or you want some other explanation here? So, since it is the first day of FDP and all of you are listening to all the speakers and even to us for the first time, so maybe some lagging is there, maybe something is not clear to someone. Somebody maybe not familiar with the platform MS Team, maybe joining for the first time. So if any issue is there, so we are having time to address those queries. So I am still waiting for a few minutes, and then we are going to end this meeting for today. If there is any query, then I am still waiting for you.
Can we leave? Dr.
Okay, yes sir, thank you so much sir, Pankaj sir. And now I would request you to kindly leave the session. Okay, in tomorrow's session, we will be having the speakers from the industries, from the academia, and from the research background. So they are going to deliver a good present, lectures on the different aspects of precision agriculture, as they are expert in the different fields related to agriculture and light, precision agriculture and light. So we are expecting the maximum presence of all the participants in all the sessions tomorrow. So since there is no query coming from your side, so I would assume that we are at the end of this third session and the last session of the day. So I would like to thank again the Pratibha ma'am for hosting this event for the day. And I would like to thank all my speakers for today's session, especially to Pankaj sir and all the two eminent speakers that we have got from in session 1 and 2. So thanks to all the participants, because without your presence, there is no means of conducting an activity. So thank you so much that you are here and you are listening to us so patiently. And we promise to bring to you more elaborated, more detailed, and more informative content on precision agriculture. Thank you so much. I would request you all to kindly leave the session, and we'll meet tomorrow. Thank you.
Hello. Hello.
Okay, yes, sir.
Sir, my question is, we need to fill a feedback form for each session.
I don't think so, sir. You need to fill it once only.
Okay. At the end of
Since at the end of like whole FDP, at the like on 24th we will be having this feedback session and assessment session. It is going to be a very short session in which we
Mhm.
can have the feedback and the assessment both. Okay.
Okay. Okay.
So.
For the attendance only means we can see from
Yeah,
that.
for attendance, again, I have instructed first also. Initially, I have also instructed regarding the same. It is on MS Team, na, sir. So automatically attendance is generated on MS Team. So once the participant join and leave the meeting, so both the times are recorded here.
Okay,
So
sir.
yeah, with that sheet, it is going to be generated in an Excel sheet form. So we will be having that record with us. So from there we can collect the attendance.
Mhm.
And like the quiz is there, so participants can also like make up their attendance by